My name is Matthew Komori, and I am a fourth year kinesiology student, and my summer research was looking at how stress is influenced by astrocyte calcium. So, understanding how specific brain cells influence behavior can help us uncover the mechanisms behind stress-related behaviors. My research was focused on a particular part of the hypothalamus called the paraventricular nucleus, or the PVN, a region of the brain involved in the stress response. So while neurons often get the spotlight in stress-related research, astrocytes, cells that actively support neurons, are being increasingly recognized as modulators of brain activity. Research has shown how astrocyte calcium can regulate corticotropin-releasing neurons. However, currently there is no research in the PVN that has looked at if astrocytes can alter this behavior through this pathway. Therefore, I designed an individual project and hypothesized that decreasing calcium in astrocytes in the PVN would increase stress-related behaviors. Using animal models, I used a viral vector to express a protein channel in PVN astrocytes. In the calyx, or experimental group, the protein channel pushes calcium out of the astrocyte, thus reducing calcium inside. The control group underwent the same procedure, but expressed a non-functional version of the protein. To assess behavioral outcomes, we tested 12 mice, 6 in each group, using 5 standard behavioral experiments: the looming shadow test, the light-dark box, the open field test, the elevated plus maze, and the tail suspension test. Today, I'll focus on the first two, where we observe the most relevant findings. In the looming shadow test, mice are exposed to an overhead shadow that mimics a predator threat. The mice with reduced astrocyte calcium, the calyx group in red bars, fled from the stimulus more frequently than the controls. This suggests that they had a heightened defensive reactivity. In the light-dark box, animals can move between a dark enclosed area and a bright exposed one. Interestingly, the calyx mice took longer to first enter the light side compared to the controls, again indicating an increase in anxiety-like behavior. So now, taken together, these findings suggest that astrocyte calcium signaling in the PVN helps fine-tune defensive responses at high states of arousal. By reducing astrocyte calcium, these regulatory effects can be lost, leading to exaggerated stress behaviors, highlighting that astrocytes are active participants in neural circuits. These findings are extremely significant because they reveal potential clinical relevance, suggesting that targeting astrocytes and their function through calcium signaling could open new avenues for treating anxiety-like and depressive-like behaviors and disorders. So, I would like to thank you, the University of Calgary, Cummings School of Medicine, the Hotchkiss Brain Institute, and Alberta Innovates for funding this research. And you can scan the QR code for my references and further data on my project.